Hello, everyone, and welcome to today's webinar titled Structure-Based Vaccine Design for COVID-19 and Other Respiratory Viruses. This webinar is part of the Microbiology Virtual Week virtual event. And I'm Susie Valdez of LabRoots, and I will be your moderator for today's event. So let's get started. Before we begin, I want to remind everyone that this event is interactive, and we encourage you to participate by submitting as many questions as you'd like during the presentation. To do so, simply type them into the Ask a Question box located on the top right corner. If you have any trouble hearing or seeing this presentation, just click on that Ask a Question box, and I apologize, on the far left corner of your screen. As a reminder, this presentation is educational and offers free continuing education credits. Click on the Continuing Education Credits tab located on the top right corner of your presentation window and follow the process to obtain those credits. I now present today's speaker, Dr. Barney Graham, Deputy Director of the Vaccine Research Center and the Chief of the Viral Pathogenesis Laboratory at the National Institute of Allergy and Infectious Diseases. For complete biography on our speaker, please visit the Biography tab at the top of your screen. Dr. Graham, welcome. You may now begin your presentation, sir.
Good afternoon. My name is Barney Graham, as you heard, and I'm going to talk to you today about the work we've done over the last 10 years or so on structure-based vaccine design for respiratory virus diseases. I work at the VRC. Vaccine Research Center at NIAID was founded in 2000 specifically to work on an HIV vaccine. And even though we have not achieved that, an effective HIV vaccine, the technologies that we've developed to make that effort have resulted in a number of other projects to address problems with unmet needs like influenza, Ebola, including a number of emerging infectious diseases. And this is made possible because we have the privilege not only to have a research building for basic research, but we have other components of the VRC, like the Process Development Group in Gaithersburg, which is a little over 100 engineers, another group in Frederick, just north of Bethesda, to work on GMP manufacturing, clinic in the NIH Clinical Center, and another group to do GLP assays on our clinical samples. And that has allowed us to explore and develop a number of delivery approaches, including nucleic acids and vectors and virus-like particles, as we did for chikungunya, proteins and nanoparticles, which I'll say a bit about, especially for RSV and the coronaviruses. I think you'll hear from Dr. Masaru Kanekio on some of the nanoparticle approaches we're taking for influenza, and a number of monoclonal antibodies, including the MAB114 that effectively treated Ebola in Eastern Democratic Republic of Congo. So we've been able to send these products around the world, either for research or clinical use, and it's done by this small group of investigators and program heads led by John Mascola, who's there in the center in the blue shirt. So I'm going to talk about not just structure-based design, but pandemic preparedness in general. And we've been thinking a lot about this during this decade, especially because of the problems we've had with chikungunya and then MERS coronavirus and then Ebola and then Zika, et cetera, and just on and on. And it's led us to consider what can we do to get more ahead of this and be more proactive. And so the approaches, including a platform approach, meaning platform manufacturing that can allow you to plug and play with vaccine antigens into the consistent manufacturing approach, or a priority pathogen approach as WHO or CEPI have taken to select the top pathogens of concern. CEPI, for instance, chose Lassa and MERS coronavirus and Nipah virus. WHO had a list of 8, but we have advocated for a more comprehensive, systematic approach that we call a prototype pathogen approach for pandemic preparedness that would address all 25 virus families known to affect humans. And the project I'll tell you about today is an example of that. We think it's possible because these new technologies have transformed vaccinology as we used to know it about 12 years ago, largely because of structure-based vaccine design concepts that I'll say more about, but also because of single-cell sorting that gives you the potential for rapid human monoclonal antibody identification, defining antibody lineages as new molecular targets for vaccine studies, and to evaluate immune responses at a single cell level in vaccine trials. It also has stimulated a whole new science on protein engineering of self-assembling nanoparticles. And these kinds of technologies have given us a new precision of antigen design and effective, to achieve effective functional responses. And the other technologies included at the bottom have helped us to go faster. And we think that combining precision with speed is what's needed for both pandemic preparedness and for pandemic response. I'll be talking more about the nucleic acid approach for vaccine delivery in this talk. So for me, this started in 2010 when we started a project to understand the structure of the F protein of respiratory syncytial virus. F is the fusion protein. It's a class 1 fusion protein analogous to the coronavirus spike, and it has an antigenic site at its apex, this red area here, that turns out to be highly neutralization sensitive and was discovered during the solution of this prefusion structure. I say prefusion because this protein rearranges to accomplish the process of membrane fusion that allows viral entry. And during that rearrangement, this prefusion molecule is triggered to rearrange into this anti-parallel 6-helix bundle post-fusion molecule. And you see this neutralization sensitive epitope at the apex is lost in this rearrangement process. So the neutralizing epitopes that were previously known were all on the post-fusion molecule and missed many of the very sensitive sites at the apex of the prefusion. When this post-fusion molecule has been used in prior clinical trials, it could only boost neutralizing activity 2 to 3-fold, which made advanced vaccine development more difficult. But if you stabilize the prefusion conformation with a C-terminal trimerization domain and internal disulfide and cavity filling mutations, you can then boost a single boost in healthy adults and achieve up to 16-fold increase in neutralizing activity, as we published just last year. So solving the atomic structure revealed this new target of vulnerability, and we now have clinical proof of concept for structure-based design and the importance of preserving these neutralization-sensitive epitopes on these prefusion molecules. And that conformational approach to vaccine antigen design, we think, can be generalizable, and we've done that now, not just for pneumoviruses, but for paramyxoviruses and now for coronaviruses. Just as an example, in paramyxoviruses, and Peter Kwong has done this now for parainfluenza virus 1 through 4. We've done it for Nipah and measles and mumps fusion proteins, and in each case shown that the prefusion stabilized conformation of these molecules is significantly, dramatically more immunogenic and potent as a vaccine antigen than the post-fusion form. So when MERS coronavirus came up in 2012 and 13, right at the time we were solving the prefusion conformation of the RSVF, we decided that this kind of approach was needed for coronavirus. We tried. Jason McClellan, who is now a faculty member at Dartmouth, worked on this by crystallography and was unsuccessful. He engaged Andrew Ward for cryo-EM to do MERS or SARS-1 spike structure, and even with cryo-EM, we were unsuccessful. But then serendipitously, we started a project on HKU1, and a couple years later asked if we could try to solve that. And if this spike was inherently more stable and allowed the cryo-EM solution to its structure, shown here. And when that structure became available and we could see side chains, Jason McClellan's group and ours started a process of finding stabilizing mutations. And at this site, at the top of the central helix that's in purple, if you put prolines there, it prevents the stacking onto the heptad repeat and largely stabilizes the fusion protein. And it turns out that those two proline mutations between the heptad repeat 1 and the central helix also stabilize MERS and SARS spike sufficiently to allow the prolines are there to allow solution of their structures and preservation in the prefusion conformation of these apical neutralization sensitive epitopes. And importantly, especially for gene-based vaccine delivery, not only did it improve antigenicity, but compared to the wild type levels of expression, the stabilized MERS spike had up to 50-fold higher protein level expression levels than wild type. And as I mentioned, it allowed us to not only solve the structures for MERS and SARS spikes, but also then endemic human coronaviruses, both another beta coronaviruses and alpha coronaviruses, veterinary coronaviruses, and even SARS-like bat-derived coronavirus spike proteins. So thinking that we had a way of making a coronavirus spike, we wanted to have a way of rapidly manufacturing a new vaccine antigen if a new coronavirus emerged, as we expected it would, since we had already had 2 events within the last 10 years. And so in 2016, after working closely with the company Moderna for mRNA, who makes mRNA vaccines, during the Zika outbreak, we made a collaboration agreement with them to do a demonstration project on the prototype approach to pandemic preparedness, using both a Nipah virus F as a prototype for paramyxoviruses and MERS coronavirus as a prototype for coronaviruses. The mRNA approach then uses a lipid nanoparticle packaging system to deliver the mRNA that's then translated into your protein of choice, depending on your overall design, that can then elicit antibody responses or even CD8 T cell responses, since it's made intracellularly. And the nature of this RNA and its manufacturing process affects the way it works. So you've heard of, there's a number of different ways of making and delivering RNA. In this case, the RNA has modified nucleosides. It's got a 1-methyl uridine that diminishes the intracellular signaling that could go through the TLR7 and 8. And it also has the double-stranded RNA cleaned up during the manufacturing process, which diminishes the innate signaling through these other intracellular sensors that would create a big interferon response. And so this RNA delivers and translates a protein that with relatively little innate signaling that allows the protein expression to go on longer. And so making the mRNA for the MERS spike, we found that either 0.1 or 1 microgram of the mRNA given twice at a 3-week interval was able to protect entirely against infection of the lung and reduce illness. And you see in the weight curves, even at the 0.01 microgram dose, which only reduced virus titers by half log, you got partial protection against disease with this partial immunity. So proline mutations in spike can stabilize it in its prefusion state. And we know that the prefusion spike trimers are more immunogenic. We thought it could be a general solution for beta coronaviruses, and we had shown that mRNA delivery was feasible. And as I mentioned, it was likely that we're going to get more coronaviruses. There's many coronaviruses in nature. It's a common virus in many mammalian species, as shown here. And over the last several hundred years, we've had 4 coronaviruses enter human population and become endemic. These top 4, 2 alpha viruses and 2 beta coronaviruses. And now over these last 20 years, we've had a SARS outbreak, then a MERS outbreak in the Middle East, and now another SARS coronavirus 2 outbreak. And it's likely that we'll have more coming from these animal reservoirs through an intermediate host, and then some of them learn how to transmit between humans. So when this new problem was reported near the beginning of January, and then around the 7th of January, we heard rumors that it may be a beta coronavirus, SARS-like virus. We became interested in planning, and at the time we were planning to take the Nipah virus mRNA into the clinic as our demonstration project. But when this happened, the CEO of Moderna, Stefan Bancel, agreed to switch this to the coronavirus and work on this new coronavirus that was emerging as our demonstration project for the clinic. Many people have pictured this virus in this cartoon version, but recently there's been a beautiful electron microscopy, scanning EM image published by Camille Eyre in the New England Journal, showing remarkable resolution. And you can then focus in on one of these viruses and see that, that it's only about 80 nanometers in diameter, including the big spikes. And there's only about 24 spikes per particle. So I would encourage people to go look at this and get a more representational virus. The important thing is the spike. And so when the sequences were released on the night of the 10th of January, and we found them on the Saturday morning, the 11th, a number of steps were taken because knowing the structure for us has been the key in developing antigen solutions for vaccines. And so collaboration again with Jason McClellan, who's now at UT Austin, our groups decided what types of constructs to order for making protein and for making probes and for constructs to make other assays. And so once we had the spike structure and we knew that it was in the right conformation, despite not having antibodies for validation, it allowed us to make diagnostic assays, as we did with the CDC. It allowed us to make B-cell probes to pull out and isolate and identify new human monoclonal antibodies, as we did with Abcelera. and Lilly, who've now got that monoclonal into phase 3 clinical trials, and to push it into vaccine development, as we did with Moderna using mRNA, but as others have done using vectors or protein-based approaches. So I'm going to show you some of the preclinical data that supports the advancement of this product into phase 3 studies. In mice, we showed that either 2 doses or 1 dose of the vaccine, followed by either 4 or up to 13-week challenges, was protective. And we did this in collaboration with Ralph Barrick, who, while we were testing immunogenicity, was developing a mouse-adapted form of the SARS-2 virus. And just by modifying 2 amino acids in its receptor binding domain, he was able to make a virus that easily infected mice. And so with 2 doses and a 4-week challenge interval, you could see that good ELISA activity was induced. The red is in the 1 microgram dose, and that level of ELISA binding activity results in about a 1 to 1,000 level of ID50 for the pseudovirus neutralization. Even at a single dose, if you use either 1, again in the red, or 10 micrograms in the purple, that you can generate good levels of neutralizing activity, in this case, again, up to about 1,000. And that level of activity from the 2-dose regimen, 1 microgram, could protect lung and upper airway from virus infection, and measured by plaque-forming units. And even the 1 dose at 1 microgram or 10 micrograms with a 7-week challenge interval was able to prevent infection in the lung. In the 13-week challenge after 2 doses, you also saw protection in both lung and nose. And in this case, also, we could show the pathology showed no evidence of enhancement. So one of the important aspects of this development program is to use sub-protective doses. In this case, the 0.1 only reduced titers by 2 logs and the 0.01 by ½ a log, to ask whether in subtherapeutic doses you generated any evidence of an enhancement of disease rather than partial immunity leading to partial protection. And so you see in the top pathology boxes that PBS group, the control group, had inflammatory nodules that happened on day 2, acute lung injury, and thickened alveolar septa and interstitial infiltrates, mostly from neutrophils and lymphocytes on day 4. And these changes were largely absent from the groups that had breakthrough infection. So partial immunity led to partial protection. We also asked about the response in aged mice, again, a 0-3 regimen followed by blood draw and then a 7-week challenge. And you see the 1 and 0.1 microgram dose groups, again, induced a robust antibody response, and that included neutralizing activity. And even this other product, which is DIV, meaning double inactivated SARS, it was inactivated both with formalin and heat, and it had, and this was made for the SARS-CoV-1 virus, and yet it still had some cross-reactive neutralizing activity in this system. And you could show that the 1 microgram in the older mice, again, protected against weight loss. In this case, the virus has not only been mouse adapted by mutation, but serially passaged and now can be virulent in older mice without any additional modification. And so in this case, the mice were protected in both the lung and the nose at the 1 microgram dose, and partially protected at 0.1. And at the 0.1, you got partial weight loss, but then recovery. The whole inactivated and PBS groups had to be sacrificed. And this is a immunostain showing viral antigen. In the top row is day 2, and in the second row is a day 4 sample. And this is a PBS control on the far left and the double inactivated, whole inactivated virus on the far right. And you see that the 1 microgram dose level, there's no virus at either day 2 or 4. There's some virus at the 0.1, partially protected animals that's largely cleared by day 4. And in both the PBS and the whole inactivated negative and positive control groups, you got a lot of virus replication and that consolidated into foci by day 4. This was associated, and the double inactivated, whole inactivated virus groups are on the far right here in the purple bars, and so this is a multiplex cytokine array, and we asked whether this positive control for enhanced disease would induce a Th2 response, and it did. You see IL-4, 5, 9, 13, and eotaxin even being induced in animals post-challenge with this kind of priming. And yet there's no indication of Th2 responses from natural infection or from any of the mRNA primed mice, including the breakthrough group at 0.1. Same is true for inflammatory cytokines. In this case, some of the inflammatory cytokines also came up in the PBS group, but they're all up and higher in this primed animal that got whole inactivated virus and alum as an ineffective vaccine. We then asked in monkeys whether these dose of either 10 or 100 micrograms of mRNA would be protective. And these are binding antibody data showing after a single dose or 2 doses that all animals responded. And if you looked at domain-specific responses like RBD or the N-terminal domain response, that you could show that at 10 or 100 micrograms, you made robust antibody responses. And they were at the upper end or even above convalescent responses found in humans with a varying degree of illness, including some people with very severe disease. So these are robust antibody responses that can be shown for both the RBD and N-terminal domains. We also looked at functional antibody responses. This is an ACE2 binding inhibition assay in which, again, at either 10 or 100, you showed responses at the upper tier or above human convalescent responses. And even a live virus neutralization assay showed responses well above what are seen in convalescent human sera. We looked at T cell responses, again, because of the association of Th2 responses with prior episodes of vaccine-enhanced disease back in the 1960s with RSV, whole inactivated virus, but also in animal models of SARS-1. And so we thought it was important to show in the humans that, in the monkeys, that peptide-stimulated PBMC showed a Th1 biased response and no Th2 responses. In the bottom 2 panels, you see on the right that also there was a strong induction of IL-21, which has been associated with T follicular helper cells, which are important for germinal center B cell activation and amplification. This level of antibody response largely prevented subgenomic RNA, which is a measure of a replicating virus in the BAL or the lower airway in the lung, compared to the control group on the left. The 10 microgram and 100 microgram dose groups had very little subgenomic RNA detectable. There's 8 animals in each of these groups. And even in the upper airway using a nasal swab subgenomic RNA, you could show most of the virus in the upper airway was replication was prevented. Next, I'll show you the phase 1 human data. In this case, there was a 0-4 week interval, and in the younger age cohort, there was doses of 25, 100, and 250 micrograms given twice that were evaluated. Some cohorts in the 56 to 70 or greater than 71-year-old age range were also immunized with some of these same dose levels. Now, in terms of reactogenicity, solicited adverse events are shown here, and there's a lot of information here, so just look overall in the first column, which shows you the responses after a single injection, and the second column shows you the responses after 2 injections, and you see there's more response after the second dose. These 3 bars represent 25, 100, and 250 microgram dose levels, and the gray is a grade 1, the blue is a grade 2, and the red is a grade 3 reaction. Almost all the grade 3 reactions are in the 250 microgram dose group. So that dose group was not continued beyond this younger 18 to 55-year-old age cohort. Otherwise, the 100 microgram and lower dose levels were well tolerated with only grade 1 and 2 reactions. The antibody responses were robust, similar to what was seen in non-human primates. In the 100 microgram dose level, it was just as immunogenic as the 250 microgram, which made it easy to select this dose level as the one that goes forward into the phase 3 trial. And so you see either for the full-length trimeric spike or on the right, the receptor binding domain specific antibody responses that everyone responded after a first dose and everyone was boosted after a second dose. And the responses are relatively well maintained out to at least 4 weeks post second immunization. And they're all at or above the convalescent sera panel shown on the far right. The same was true for neutralizing antibody that was measured both by pseudovirus neutralization or by classical plaque reduction neutralization, PRNT, with live virus. And again, the activity was at or above in the upper quartile of convalescent responses. In this case, there was some neutralizing activity seen after the first dose, but all people responded, 100% of responders after the second dose. And even in the elderly cohort, I'll show you just some of this unpublished data that indicates that at 25 dose levels, 25 microgram dose levels in the younger age, the 56 to 70 and the greater than 71 on the 3 groups on the left, all responded in a similar to a similar level. And at the 100 microgram dose level, the older 2 age cohorts of 56 to 70 and greater than 71, if anything, were at or above where the younger adult cohort was. So responses were also robust in elderly subjects at the 25 microgram and 100 microgram dose levels. Again, the T cells were done as looking for a safety signal, and in this case, there was no Th2 response, and the response was heavily biased toward Th1 cytokine production, which is what we were looking for. And there was some, but very little CD8 T cell response detected in the peripheral blood. So in summary, both in mice and monkeys and humans, we have immunogenicity data supporting this approach. We have protection data in mice and monkeys in both upper and lower airway. And we have safety data established in humans that all supported advancing into this phase 3 trial. And so this was all done in very rapidly. And so as I mentioned, we were waiting for the sequences to be released on the 10th, and we got them on the 11th. We ordered the reagents we needed to make protein for solving the structures and making the assays and isolating new antibodies. But we also selected a sequence based on our initial MERS work, putting the 2P mutations, 2 prolines into the 986, 987 amino acid positions between the heptad repeat 1 and the central helix. And because of that prior experience, because of our prior relationship, Stefan Bonsell was willing to, at risk, make this decision to go forward with manufacturing and allowing a GMP product to be available by 41 days after sequence released. And within just a few more weeks, the phase 1 trial was able to start within about 65 days of the sequence release. There's now a phase 2 trial that started in May, and the phase 3 trial, as I mentioned, started July 27th. And even though this was rapid, I want to emphasize that not many steps have been skipped, if any, in this process. The things that have been put at risk were money and time. And we had non-clinical immunogenicity data before starting phase 1. We had protection data in 2 animals before starting phase 2. And we had safety data by limiting dose of vaccine and breakthrough infection and pathology and immunology before starting phase 3. And now we're in the midst of a phase 3 trial that is more than 2/3 enrolled. And this is not the only product going forward. There's mRNA in these and in phase 3, both from Moderna and from the Pfizer-BioNTech collaboration. There's whole inactivated virus vaccines from China, and recombinant advectors from the UK and China, and also coming along quickly are recombinant subunit proteins that appear to be very immunogenic. And so there's 34 vaccine candidates in clinical evaluation, and 7 of them have already reached phase 3 trials. And according to WHO, there's at least another 142 vaccine candidates in preclinical evaluation, including things that use nanoparticles, chimeric viruses, additional vectors, and virus-like particles. So there are a lot of options here, and it's remarkable what's happened this last year in terms of the promise of vaccine technologies really coming to the fore in this crisis situation. So these are some of the products that are advancing. As I mentioned, Moderna and Pfizer both have RNAs that are already over 2/3 enrolled in phase 3 trials. AstraZeneca just recently started their phase 3. Novavax and J&J will be starting soon. And so there's a lot of progress being made. And I just put asterisks by these products because 5 out of the first 6 products, at least in phase 3 in this country, are using the 2 proline stabilizing mutation as what they considered maybe a better vaccine antigen. This is all being organized under a new system that was designated Operation Warp Speed, but it really is a way of more of organizing and with all due deliberation the multi-agency effort in the government combined with the multi-company effort from all the pharmaceutical companies. And so this is not only covering vaccines, but therapeutics and diagnostics. The vaccine group has both components of vaccine development, supply and manufacturing, and also a big work on delivery and administration. And just to show you how the government approached this problem, the prior systems, including the HIV Vaccine Trials Network, the HIV Vaccine Preventions Network, and the old VTU, and now the new IDCRC, and even DoD sites combined into a consortium of government sites now called Coronavirus Prevention Network that covers much of the country, as shown here in these red dots. And when you combine that with the company's CRO sites that are contract sites, you can cover virtually the entire country. And in the case of Moderna, there's 99 sites activated to enroll this trial. There's a website now for the network if you're interested in finding out more about it, coronaviruspreventionnetwork.org. And there's a big effort through DoD and others and HHS putting predictive analytics and really amazing modeling and computer power behind this to try to anticipate how the outbreak is moving by using county maps of prevalence and new cases and overlaying that with the testing sites and places like meat and packing companies who have known to have major outbreaks and now colleges, et cetera. So these kinds of approaches will help shift emphasis on enrollment to try to capture cases in the phase 3 trial. So I'll summarize here by just restating that this is all made possible. The rapid development was made possible by prior fundamental, basic, and translational research. This idea of combining precision vaccinology and platform manufacturing for both precision and speed. Proactive planning toward virus families that may have a proclivity for pandemic threats, and pre-established public-private partnerships, which allowed this to go rapidly. And this mRNA 1273 is well tolerated in mice, monkeys, and humans. It's protective in mice and non-human primates. It's immunogenic in older age groups. And as I mentioned, the phase 3 trial now is more than 2/3 enrolled. And I just want to thank the PIs that I introduced earlier that I work with every day. Jason McClellan, who's now a professor at UT Austin, is still an important collaborator on many things. Dr. Kizmikia Corbett, who's hiding here, has been the leader of our small coronavirus sub-team and has generated much of the data that you saw on MERS and the early data now on this new virus. Our collaborators at Moderna, Ralph Barrick and his group at UNC, who's done all the mouse challenge studies and live virus assays, and Mark Dennison at Vanderbilt and Jim Chappell, who have done a lot of the PRNT work and have been both long-term collaborators. So I will stop there and take any questions if you have them.
Thank you, Dr. Graham, for that informative presentation. And we will now start the Q&A portion of this webinar. If you have any questions you want to ask, this is the time. Please do so now by clicking on that Ask a Question box located on the far left of your screen. So let's get started. We already have a couple questions coming in, and we hope to get some more, Dr. Graham. Our first question is, at what stage of enrollment is the phase 3 study?
Yeah, so we are around 21 or 22,000 in the Moderna trial, and I think the Pfizer trial, which is using an mRNA and a stabilized spike prefusion trimer, very similar products to the Moderna. They're up well over 23 or 24,000. So these trials, which have a target enrollment of 30,000, should be there within the next few weeks.
Thank you so much. And our next question, there's been a lot of concern about the vaccine safety. What steps have you taken to ensure that the vaccine is safe?
Yeah, many people just are inherently distrustful of vaccines, and because of this going so fast and people not really understanding why it was able to go so fast, there's been a lot of concern about maybe it's going too fast and it's not safe. But I hope that I explained to you some of the basic biology and scientific underpinnings of why it was able to go faster this time than it has ever gone before, some of the pre-established relationships and information we had. And that we have generated a lot of data, both in animal models and in humans, and have gone through all the typical phases, but it's just all done in parallel. And at the same time, a lot of risk is being taken on advanced manufacturing and process development for these different products that typically would be done serially instead of in parallel. So a lot of money has been put at risk, but I hope I've been able to convince you that none of the essential steps in safety have been skipped. And in fact, in some ways, they've been scrutinized even more heavily because of this speed than we often do.
Thank you so much. Dr. Graham, we have time for one more question. I want to give you some closing remark time as well. When will you know the answer from the phase 3 study?
These are endpoint-driven trials. So the second doses in at least the Moderna trial started a few weeks ago. And so 14 days after that second dose was September 7th. So we've only been counting new cases, symptomatic cases that are PCR confirmed, starting from the 7th of September. And the first interim analysis will be when we get up to 53 cases. The next analysis we think that could give an answer would be at 151 cases. So it really is a matter of infection rates and where enrollment has happened and who gets infected and who isn't, and potentially how effective the vaccine is. If it's highly effective versus modestly effective, it would affect the timing. So that's going to be an experimental question.
Dr. Graham, this has been a great presentation. Thank you so much. Would you like to provide the audience with some closing remarks before we end our presentation today?
Well, I think the most important thing that I think this work represents is the importance of basic research in being prepared for future problems, because it was really knowing the structure and knowing how to stabilize structures that at least allowed our group to get off to a fast start. And so the importance of basic research just being underneath all of the activities that have happened. The prior planning and prior relationships, I think, are critical. And the idea that we could potentially, with some relatively minor investment, which seems minor, especially considering all the trillions of dollars that are being lost out of our economy now, that could be invested in proactively studying and preparing for potential pandemics out of the other 25 virus families that we know can infect humans. And the efforts that would be spent on solving structures and making antibodies and developing animal models and things like this, I think would put us in a much better position in the future for unexpected outbreaks.
Dr. Graham, thank you again for your presentation today. Before we go, I want to thank the audience for joining us today and for their interesting questions. And any questions we did not have time for today and those submitted during our on-demand period will be addressed via the speaker of the contact information you provided at the time of registration. And this webcast can be viewed on demand for 6 months until March 2021. LabRoots will alert you via email, when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, be safe, stay healthy, and take care. Bye-bye.